This is the fifth and last video of the second lecture for ionotropic receptors. In this video, we will focus on discussing purinergic receptors. Purinergic receptors, also known as purinoceptors, are a family of transmembrane proteins found in almost all mammalian tissues. These receptors have been implicated in learning and memory, locomotor and feeding behavior, and sleep. And they can be found in pre- and postsynaptic nerve terminals, as well as glial cells through the central, peripheral, and autonomic nervous system. These purinergic receptors are responsible for a fast excitatory response in both neurons and muscle. They bind ATP, and there are two different types of purinergic receptors, ionotropic and metallotropic. And for this video, we will discuss the structure and function of the ionotropic purinergic receptor P2X. As I just mentioned, they bind primarily ATP, but they can also bind adenosine. And unfortunately, their primary structure has not been determined yet, so we lack a lot of knowledge on where things bind and the different amino acids that will be essential for the activation of these receptors. However, homology studies have shown that they have similarities to the potassium channels. This is a branch of how different purinergic receptors are classified and the different types of the purinergic receptors. As I just mentioned, purinergic receptors are responsible for a fast excitatory response in neurons and muscles. As we briefly saw in the first lecture of the ionotropic receptors, these receptors are composed of three subunits represented by this diagram. So three different subunits come together to form the receptor. And every subunit is formed by two transmembrane domains, TM1 and TM2. In this type of receptors, the N-terminal and carboxy terminal domains are facing the intracellular space, while the ligand binding or extracellular domain is in the outer membrane. These purinergic receptors have also different subunit compositions, and also their diversity is reflected by the assembly of the different subunits to form these trimers. So purinergic receptors can react to changes in the ATP extracellular concentration. And levels of ATP in the extracellular space can be changed by different phenomena. Those can be affected by exocytotic ATP released into small vesicles that are released into the extracellular environment. And then microvesicle mediated ATP release. Once they are outside of the cell, these vesicles break down and release the ATP content. They can also be mediated, or they can be exchanged by a connexin pannexin 1 ABC cassette or pump that pumps out ATP into the extracellular space. So there are different mechanisms that can really change the levels of ATP, and purinergic receptors can sense those levels and respond to that. Then here in this scheme, we have a diagram of the this trimeric complex for the P2X ionotropic purinergic receptor. And this type of receptor allows sodium and calcium to flow inside the cell and allows potassium to go outside. And as usual, I leave you here with a short quiz where you can revise the most important concepts that we've seen in this video, and we will discuss these and other questions in class.